Okay, so this is the fourth and final video for the digestive system. All right, so we left off last time. We were going to talk about the pancreas. All right, so the pancreas, so far as histology of the pancreas, we mentioned this before when we did the endocrine system, because it is both an organ of the endocrine system as well as the digestive system. So when we look at the histology here, right, it's actually made up of these structures, right, these secretory structures called acini, right? So these are, if I were to kind of, let's see, let's just circle one. All right, that would be a serous acinus, and that's what we're interested in here with the digestive system, because that's what's going to be making those digested juices. But these serous acini surround these islets of Langerhans, right? And they secrete that insulin and glucagon, those hormones. So, but for the digestive system, all of our digestive juices for the pancreas are coming from these serous acini. So let's talk about that pancreatic juice. Okay, so the pancreatic juice, as we mentioned before, it consists of both an alkaline kind of buffer substance as well as a really enzyme-rich juice, right? So there are two kind of different juices. Generally, they're going to be secreted at the same time, all right? But there's definitely two kind of significant components. So we have this alkaline buffer, all right? This is going to be full of bicarbonate, and that's going to be stimulated by the hormone secretin. Okay, so then we have this really enzyme-rich juice coming from the pancreas, stimulated by CCK. And remember that CCK is activated by the presence of fats and carbohydrates in the duodenum. Okay, and so it's going to have a bunch of enzymes to digest pretty much every kind of macromolecule. So let's go through those. Okay, so let's first talk about carbohydrate digestion. So for carbohydrate digestion, the pancreatic juice is going to contain pancreatic amylase, right? And we've seen amylase before in saliva. So this is just an enzyme that digests carbohydrates. Okay, and so then for protein digestion, remember, protein digesting enzymes are secreted as proenzymes. That's just a protective form so that they don't digest anything around the cells that are secreting them, and instead digest, the protein they're supposed to from food. So there's three different protein digesting enzymes that you should know. So the first one is trypsinogen, right? That's the proenzyme, and it's activated into trypsin. Okay, and so it's activated by another enzyme called enterokinase. And so enterokinase is an enzyme embedded in the wall of the duodenum. Okay, so this proenzyme trypsinogen has to kind of rub and kind of come into contact with the wall of the duodenum before it can be activated into trypsin. And that actually makes a lot of sense if we look for a second before where we had our picture of the pathway. All right, actually, let's go down. I think I have a picture of the pancreas here. Here we go. All right, so this in the pancreas, right, the actual pancreatic duct here is very long. Okay, and that protein digesting enzymes can be made, right, anywhere out here and added to that pancreatic duct, and then it has to travel before it reaches the duodenum. So having one of the kind of means of activation for this being that the trypsinogen has to actually get to the duodenum would make a lot of sense. So it's only active once it actually reaches the duodenum. Okay, so that's trypsinogen activated to trypsin by enterokinase in the wall of the duodenum. And then that trypsin, once it's activated, will activate our other two enzymes. So the first one is chymotrypsinogen. It's activated to chymotrypsin. And then procarboxypeptidase is activated to carboxypeptidase. And so both of those are done by trypsinogen. So again, they can't be activated until they're in the actual duodenum where they need to be digesting materials. All right. Okay. So then lipid digestion is going to be done by the enzyme lipase. Okay, and so remember, we also have bile being added into the duodenum at this point from the gallbladder or the liver. So those bile salts are going to help emulsify that fat, so this lipase can more efficiently break down those little fat droplets. Okay. And then lastly, we have enzymes in pancreatic juice to digest nucleic acids. All right, so this is DNA and RNA. The enzyme that digests DNA is called deoxyribonuclease, right? That digests deoxyribonucleic acids. And then we have ribonuclease, which digests RNA, right? Ribonucleic acids. Okay, so those are the enzymes. And then if we just follow the pathway of pancreatic juice, it is going to go from the serosacini that make it out here, right, into the pancreatic duct, which is here. All right, that pancreatic duct is going to join that common bile duct, okay? And then they will, once they join, right, they go to a structure, I'll kind of draw it in here, called the ampulla of Vater, right? It's just like an enlarged kind of ampule. And then there is a sphincter of Oddi. I'm just going to draw it as like a little circle here. It's like just a little ring of smooth muscle that makes a sphincter that controls, you know, when that juice is secreted into the duodenum. All right, so it would travel through that sphincter when it opens. Okay, good. All right, now let's talk about nutrient absorption in the small intestine. All right, so nutrients absorbed in the small intestine must travel through the intestinal cells in the small intestine. They cannot travel between the cells because, remember, they're held together by tight junctions. So there's no way for those nutrients to just move between cells. So they have to go through an actual cell. And so that means what they're going to do is they're going to enter, all right, on this apical side. So this is the side that's going to be facing the lumen. Okay, so if we're just using glucose as an example, right, it'll enter that apical side, all right, and then it has to travel through the actual cell and then leave the basal side in order to enter the blood vessel, which is actually its, you know, final goal, right, is to get into the blood so then it can be transported to the liver. Okay, so let's talk about specifically, right, this process here. Okay, so how do these nutrients get into that intestinal cell? Okay, and so that's going to be accomplished by a symporter. And so a symporter is just a membrane protein that transports two molecules, right, at least two molecules into a cell in the same direction. Okay, and so these nutrients are going to be transported along with sodium. Okay, so it'll be like glucose with sodium or amino acids with sodium. Okay, and so the reason they're transported with sodium is because sodium has that really high extracellular concentration that's created by the sodium-potassium pump. All right, so that sodium-potassium pump, remember from, you know, skeletal muscle and the nervous system, is constantly pumping out sodium, all right, and pumping in potassium. So we have a really high concentration of sodium on the outside of the cell. And what that means is when this symporter here opens, there's almost like this like whoosh, right? Like this waterfall, this current of sodium rushing into the cell. And so what the amino acids and the glucose can do is they can kind of just hitchhike along that current, right, and make it into the cell themselves. That's kind of how it works, okay? So really this, aside from the ATP needed for that sodium-potassium pump, this process here does not require ATP because you're just, these nutrients are using that concentration gradient of sodium. Okay, so then glucose would move into the cell, right, with sodium. All right, and so then we would end up with our glucose in the cell, and that sodium can be pumped back out by that sodium-potassium pump. And then our amino acids, right, are going to hitchhike along with sodium. So now we have our amino acids inside the cell. And so then they can move through the cell and then out the other side to the blood. Okay. All right. I think that was it for those. Okay. And so that's specifically for glucose and amino acids is how they're transported. However, with lipids, as I mentioned before, it's much more complicated, and that's because lipids are nonpolar, right? And so they're not going to get along with sodium, right? They're not going to, use like a membrane protein to get through. Actually, once something that is fat-based reaches the membrane, it can actually just move, because the membrane is mostly fat, it can just move straight through the membrane. It's reaching the membrane that's going to be the challenge, all right? And then coming out the other side, right, is also important too, because it'll then be traveling in a fluid environment, so it needs to be protected from that as well. Okay, so let's talk about that. So lipids for absorption, all right, we said are emulsified by bile, all right, and then they're digested by lipase. And so when they are digested, our triglycerides are actually, instead of breaking it down completely into our 3 fatty acids and our glycerol, they're broken down into 2 fatty acids and a monoglyceride. All right, so I've kind of drawn that down here so you can see what I'm talking about. So our normal kind of triglyceride structure is that we have this glycerol backbone and we have 3 fatty acids on it. Okay, so once those have been digested by that lipase, it leaves a monoglyceride, so glycerol with 1 fatty acid attached, and our 2 fatty acid chains. Okay, and so what's going to happen is those are going to be packaged up by the bile salts. All right, and so remember those bile salts from last time have a polar region right here and a non-polar region right here. They're amphiphilic. And so they're going to point that non-polar region towards those fatty acids and the monoglycerol. And then they have that non-polar region facing out, kind of protecting everything from that watery environment. Okay, so this structure with the bile salts is called a mycele. Okay, and so myceles are going to be bile salts, and they're going to be surrounding the fatty acids, the monoglyceride, and then also packaged in there is going to be cholesterol and other, fat-soluble vitamins. All right, are going to be packaged in there. So you can see in here, right, we have our cholesterol and our fat-soluble vitamins in addition to our actual parts of our triglyceride. Okay, so that's how it's kind of all packaged up in the lumen of the small intestine. So now these myceles, all right, are going to approach our apical surface of our columnar cell. All right, so this is our apical surface. And remember, because they're made up of fats, they can simply diffuse into the apical side of the cell, right? The membrane of the cell is made of fats, and so it gets along just fine with those lipids. And so they can just diffuse right into the cell. They don't need a protein transporter. Once they're in the cell, they're going to go to the smooth ER, right? That's what we have here. They're going to go to the smooth ER, and that smooth ER is going to reassemble that monoglyceride and those fatty acids into a triglyceride. And then from there, all right, that triglyceride is going to be wrapped up in proteins. Okay, so this is kind of the second packaging to protect it on its second voyage out into a watery environment. So when it leaves the cell again, it's going to leave and go into a mostly serous solution, and it's going to need packaging. Okay, protective packaging. So in this case, bile salts were our first choice for our mycele, but in this case we're using proteins. Okay, so we call that, when we take a fat and we wrap it in proteins, that's called a lipoprotein. This specific lipoprotein is called a chylomicron. All right, so it's these triglycerides wrapped in protein. All right, those proteins will protect it from water, right? Make it water soluble. Okay, so then our little chylomicron here, right, that's made in the smoothie R of our columnar cell, is going to leave the basal side of that columnar cell, and what it's going to find is a lacteal. Okay, so it's going to go and find that lacteal in the villus of the small intestine mucosa. Okay, so it'll find a lacteal, and it will enter that lacteal. Remember, these are lymphatic vessels, so they're going to contain lymph, all right, which is mostly water. And so they're going to travel through that lacteal, through other lymph vessels, until they reach the jugular vein, and then they can just enter general circulation. Okay, so that is the kind of life cycle almost of lipids, right, that are in your food, and then how they end up in your blood, and your body can use them from there. Okay, so what I would recommend is kind of practicing this as honestly a story, right? So what happens to our triglyceride, right? Maybe you can even write it out in steps, right? First, it's broken down, packaged up with bile salts into a mycel. It enters the cell by diffusion. It's reassembled into a triglyceride, packed up with proteins as a chylomicron. That chylomicron leaves the basal side of the cell, enters a lacteal, travels through the lymphatic system to the jugular vein, where it joins the general circulation. All right, so write out some steps and try to learn it as kind of a story, a process. Okay, so let's continue with absorption. So in addition to fats, that was fat absorption, there's a lot of water absorbed in the small intestine. So 90% of the water from the food we eat is going to be absorbed in the small intestine. And that's because all of that water is going to follow the sodium through those symporters. Okay, so water is going to be absorbed along with the glucose and the amino acids. All right, and then also in the small intestine, we're going to be absorbing vitamins and minerals. So these vitamins and minerals are not digested, right? They're just absorbed as is. There are going to be fat-soluble vitamins. Remember, we talked about those being packaged in the mycels. And so those are generally referred to as the K'd vitamins, because the fat-soluble vitamins are vitamin K, vitamin A, vitamin D, and vitamin E. Right, so those are the K'd vitamins. B12 is another vitamin we mentioned is absorbed from food, and you need that intrinsic factor made in the mucosa of the stomach in order to absorb it. And then minerals that we'd be talking about would be like sodium, potassium, and calcium. All right, so now let's move on to the large intestine. So the last segment of the digestive system. So just to point out a little anatomy of the large intestine, all right, so the ileum here, right, will join with the large intestine at what's called the ileocecal valve. That's between the ileum and this structure here, which is the cecum. All right, so that's the first segment of the small intestine, and then you can actually see here coming off of the cecum is the appendix. All right, and as you know, the appendix can be removed, although it's been found out more recently that it actually does have some kind of immune functions and lymphoid functions as well, but it can be removed without, any real problems. If it becomes inflamed, you get appendicitis or something like that. Certainly removing it is better than it rupturing from appendicitis. Okay, so then from the cecum, the large intestine goes to the ascending colon. All right, so that's going to be here. So it's going to be traveling, so if I were to draw, right, the pathway We go through the ileocecal valve there into the cecum, travel up through the ascending colon, and then it'll travel across through the transverse colon. All right, and then it would travel down through the descending colon. Okay, and so then we travel down here, and then the next stop will be this segment of the colon called the sigmoid colon, and that will lead to the rectum. All right, so then this would be here is the rectum. Okay. All right, so let's talk about the histology really quick of the large intestine. So with the mucosa of the large intestine, it's going to be just like we talked about with most of our organs, simple columnar with lots of goblet cells. And then the only other really significant thing you should know about the histology is for the muscularis externa. So with the muscularis externa, it has a special structure called the tinea coli. And so the tinea coli is where they take that outer longitudinal layer of the muscularis externa, and instead of it fully wrapping around the entire large intestine, right, all across its length, it's collected into this, and that's what this white line is here, this single band of smooth muscle. Okay, so instead of wrapping all the way around, we take that and we smush it all together, right, into this single band. And that's the tinea coli. Okay, and so when the tinea coli contract, they form these structures called haustra. All right, and so that's these pouches. So you always see kind of this pouched structure to the large intestine. These are all haustra, right? Those pouches in the large intestine. Okay, and that's just a feature of that muscularis externa. Okay, so physiology for the large intestine is primarily going to be water absorption. So 9% of the water from food is absorbed. That makes a total, right, of 99% of the water is absorbed in both the small intestine and the large intestine. All right, so there's about 1% of water left in fecal matter. Okay, so aside from absorbing water, essentially the job of the large intestine is to move that fecal matter out through the rectum. Okay, so there's a few movement-based processes we'll talk about. The first one is the gastroileal reflex. So this is that initial movement of the material from the small intestine into the large intestine. So it's going to move material from the ileum, right, through that ileocecal valve. We can add that in here, right? Ileocecal valve, all right, into the large intestine, right, the cecum. Okay, and it does that by peristalsis, right? So the small intestine moving that material forward. And so then once that material reaches the large intestine, all right, that is obviously going to stretch the large intestine. All right, there would be stretch receptors, and that's going to stimulate haustral churning. Okay, so those haustra are just going to kind of contract and churn, and that'll help to squeeze that last 9% of water out of that material. Okay, so then the last type of movement involved with the large intestine is called mass movement, or sometimes called mass peristalsis. Okay, and so this is the movement that forces fecal matter into the rectum. Okay, so it's like one big contraction that moves fecal matter into the rectum. And so really quick before we talk about that defecation reflex that's triggered by that mass movement, let's just talk about feces for a second. So what's in feces? So obviously it's going to contain a lot of undigested materials or undigestible materials. frequently, you know, plant matter, certain kinds of plant matter with a lot of cellulose in it is going to be very undigestible. It will also contain that bilirubin from the bile. That's actually what gives feces its kind of brown color, is from that bilirubin. All right, so remember that's a byproduct of heme breakdown. And then also there will be epithelial cells in feces, and that's just from the movement of the feces through the intestinal tract, right? It'll be picking up epithelial cells all along the way. Okay, so then now our last thing to talk about is the defecation reflex. All right, and so this defecation reflex is triggered because normally the rectum is going to be empty. So when mass movement or mass peristalsis occurs, right, it's going to push the feces into the rectum, and that stretch of the rectum is going to signal that defecation needs to occur. That signal is sent through the sacral spinal cord. This is a reflex, right? So it just goes through the spinal cord and comes back, and that signal triggers the internal anal sphincter to relax. All right, that sphincter is made up of smooth muscle, so that process is completely involuntary. All right, and that causes the urge to defecate. And so then the last process of defecation is voluntary, right? That's the opening of the external anal sphincter, and that's made up of skeletal muscle, so you know we do have control over that. All right, so that is it for the digestive system. That was a really big chapter, a lot of information. So study, right? Remember your enzymes. You should know where they're made, what they're made to digest, right? And where they're acting. And then the hormones, you need to know what cells are making them, why they were released, what tissues they're acting on, and what their effects are. All right. And so then I will talk to you guys in our next video.